Hello everyone, I'm Mike Sierk from the Center for Biomedical Informatics and Information Technology at the United States National Cancer Institute, and I'm going to present a new tool that my colleagues and I built called 3DVizSNP. The tool is available as a Python script on GitHub or as a web server hosted by NCI. If you have problems running the program or have suggestions to improve it, please raise an issue on the GitHub repo or send an email to the help page of the web server. This work was also recently published in BMC Bioinformatics if you'd like to get more details there. The problem 3DVizSNP is addressing is evaluation of the phenotypic impact of missense mutations on protein structure and function. High throughput experiments generate large numbers of variants that must be prioritized for likely phenotypic impact on protein structure and function. There are numerous tools available for sequence-based scoring of missense mutations. But ultimately, one must have detailed structural and functional information about a protein in order to predict with confidence what the effects of a particular mutation will be. Typically, this information is expensive and time-consuming to generate, with only a small fraction of the available sequences having high-resolution structures available in the PDB. However, the new structure prediction algorithms such as AlphaFold have greatly expanded the number of proteins with high-quality structural information available. At the end of the analysis pipeline, there are algorithms that can produce detailed biophysical calculations or simulations on mutants where the structure is available. But this is generally computationally expensive and requires specialized knowledge to implement and interpret. As illustrated in Figure 1, 3DVizSNP is meant to fill the gap between easily accessible sequence-based scoring of mutants and more difficult to produce detailed structural analysis of mutants. It allows researchers to efficiently gain insight into the structural context of a mutation without requiring high computational overhead or extensive levels of structural expertise. Figure 2 depicts the 3DVizSNP workflow. It requires a variant color format file as input, which is then processed to extract missense mutations, with the option to restrict the variants to a list of gene symbols of interest. The variants are submitted to the REST API of the Variant Effect Predictor server hosted by Ensemble. The result is parsed in a data frame created with the variant, the Ensemble gene ID, gene symbol, UniProt ID, and the SIFT and polyphen predictions and scores. The program then identifies if a PDB structure is available for each mutation using the PDBE REST API, correcting any numbering offsets between UniProt and PDB, and also avoiding structures that have an engineered mutant at the site in question. Then a URL is generated for loading the mutation in the ICN3D web-based structure and sequence viewing software, which was created by two of my poster co-authors, Jiao and Philippe. It will either pull up the PDB structure if one is available, or the AlphaFold 2 predicted structure for proteins under 2,700 amino acids. The whole data frame is exported as an HTML page and a comma-separated values file. The web server output is shown in Figure 3. The table of variants is sortable and filterable, and the full ICN3D web page can be opened by clicking on a button. The selected variant row is highlighted in green, and the mutation is displayed at the top of the page in an embedded version of ICN3D. Clicking on a new row brings up that mutant in the embedded viewer. In addition to the 3D display, ICN3D has a 2D contacts window displaying changes upon mutation, and a 1D sequences and annotations viewing window. ICN3D uses the scap command to allow a user to toggle between wild type and mutant versions of the amino acid. This is illustrated in the animation in Figure 4. In Figures 5 and 6, we demonstrate the use of 3DVizSNP. In Figure 5, we use the -G flag when running 3DVizSNP on a VCF file from a colorectal cancer patient from TCGA. This allowed us to select only the variants from the KRAS kinase protein. The output included the well-known glycine 12 to valine mutation, shown in panel A, which affects the active site of the enzyme. And another mutation, aspartate 154 to asparagine, shown in panel B, which affects a hydrogen bond at the dimer interface of the protein. In Figure 6, we started with a VCF file from a glioblastoma patient from TCGA that had 27,000 variants in it. We filtered the file down to a list of 900 variants using the Open Cravat server, which we then ran through 3DVizSNP. Going through the 3DVizSNP output table one at a time, we identified a leucine 194 to arginine mutation in a cytochrome P450 enzyme. We submitted this mutation to the missense 3D and ISMB web servers, which both predict that the mutation is deleterious to protein stability. We further found that there is a link to the DisGenet Gene Disease Association database on the UniProt page for this protein, which lists an article that claims expression of the protein is protective in liver cancer. We were thus able to use 3DVizSNP to quickly identify a mutation that has evidence supporting that it could be relevant to the progression of glioblastoma. We believe the 3DVizSNP tool makes it easier to rapidly visualize mutations generated in high-throughput experiments in their 3D context. We hope you find this tool useful. Thank you for listening to this presentation.